Dutch, and thanks for the invitation to give this webinar today. So today, as Katrina said, we're going to be talking about some wettability work we did on unconventional samples. This is a work we did it's been several years now, but we kind of started before COVID and continued during COVID. We haven't done too much recently, but it's work that people find interesting and we like to share. It really showcases the capability of NMR. So that's what we're going to talk about today. So as just a quick introduction, as everybody knows, wettability is the affinity of a surface, of a fluid to a surface. And it can, in rocks, it can depend on many things, including the mineral composition of the surfaces, the composition of the in situ brine, the complexity of the oil chemistries, and the maturity of the organic matter in the pore volumes. And these are all characteristics of unconventional reservoirs. So this makes determining the wettability of unconventional reservoirs difficult. And as you probably all know, there's currently no industry-established core analysis wettability evolution methodology for unconventional resources. Traditional methods like the AMOT or USBM methods that use for conventional samples aren't really useful or can't easily be applied to unconventional samples. This is both because the tight rocks of unconventional samples have small volumes, making quantifying saturation changes difficult, and there's limitations of the conventional instrumentation. For example, pressure pushing fluids through these samples can be difficult due to their low permeabilities. And for these reasons, over the past several years, we've developed a T2-based NMR wettability index for measurement of the wettability of unconventional samples and to provide a robust wettability measurement for tight rocks. So before I get into the specifics of the wettability measurement, I just want to spend a couple slides reviewing what the T2 measurement is and why it's important to NMR core analysis. So as many of you probably know, T2 decay or transverse relaxation decay is probably the most important measurement we do in NMR core analysis. And this is because a T2 relaxation is proportional to the pore size distribution and the porosity in these samples. For example, the transverse decay is given by this equation right here. And when the rock is saturated with only one fluid, then it's proportional to the surface to volume ratio of the pore volume of the sample. And this is because T2 bulk is typically large, so 1 over T2 bulk is typically small, so we can somehow safely neglect this term. So then T2 is inversely proportional to the surface to volume ratio. This means that the transverse decay of small pores will have short T2 relaxation times, whereas large pores will have long T2 relaxation times. And then what we typically do is when you measure all the relaxations at once. We invert it from the time domain to the T2 domain, and now you have a pore size distribution where it shows how much fluid is in each of the pore networks. And this is true, like I said, for samples that are saturated with only one fluid. But in mixed saturation samples, like the ones we're going to talk about now in this study with our wettability measurements, it's not just the surface to volume ratio, it's also this rho term or the surface relaxivity term. And this term should be somehow connected to the wettability. So in a sense, we should be able to, in theory, use these T2 distributions to tease out the wettability. And that's exactly what we've done in this study. So before I start talking about the specifics of our measurements, of our wettability results, I'll talk a little bit about how we measure the T2 relaxation. Well, we use a Carr, Purcell, Mayboom, Gill NMR pulse sequence. And briefly, what this sequence is it takes all the nuclear spins, which are initially aligned with the magnetic field, and we apply what's called a 90-degree pulse, and that excites all the nuclear spins so that they're pointed 90 degrees perpendicular to the direction of magnetization. These are now excited, so they begin to decay, and that decay is the transverse relaxation decay. Then after a time tau, if you apply a 180-degree pulse, it refocuses the nuclear spins and causes an echo at a time tau later, tau after the 180, and then you get an echo, and you get your first echo, and that you continue to do that over and over again with these 180-degree pulses, continuing to refocus the echoes. But the echo size does decay slowly over time, and that decay is the T2 relaxation time. And that's the time, as I said, we're interested in to tease out the wettability, or in normal, most of the time, to get the pore size distribution. So, okay, so let's get into some wettability measurements. So here you go. So here is the 100% of a typical sample. Here's the 100% water saturated, 100% crude oil saturated, T2 distribution, blue and yellow. Red is the 100% is the T2 distribution of bulk brine. Green is the T2 distribution of bulk crude, and the brown trace here is the measured T2 distribution of a sample, the same sample that the 100% water saturated and 100% crude saturated T2 distributions were recorded for. Only this time for the brown, it's mixed. It's got oil and water in it, and its T2 distribution looks like this. So the question is, can you mix the blue, the yellow, the green, and the red T2 distributions together to give the brown T2 distribution? And if done correctly and quantitatively, can you get the wettability out from this mixing? And the answer is yes. And I just want to be abundantly clear right here from the beginning. We didn't develop this T2 wettability measurement. It was Looyestijn and his co-workers who were instrumental in developing this technique. All we've done is further this work and show that his technique is capable of monitoring the change in wettability for unconventional samples. So I'm now going to spend several slides trying to explain this NMR wettability model and how we mix these T2 distributions, the blue, the yellow, the green, and the red, together to get the black. Well, the model at its most simplest says that there are two types of pores in a sample. There's pores that are wetted by water, and there are pores that are wetted by oil, shown in this figure here. So then that means, the consequence of that means is there's four different types of fluid in any core sample. There's water on the pore surface of the surface of the pore, oil on the surface of the pore, and hence there's oil in the middle of the pore, of the water wet pore. In the middle of that pore, there's oil, and in the middle of the oil wet pore, there's water. And so the T2 distributions that I just showed on the last slide are each of these four. So for water on the surface of the pore, we say that will look like the 100% water saturated T2 distribution, whereas oil on the pore surface will look like the 100% oil saturated T2 distribution. And then oil in the middle of the pore of a water wet pore should look like bulk oil, and oil in the middle of a water wet pore should look like bulk oil, and water in the middle of an oil wet pore should look like bulk water. And then this is the equation for the NWY model. And then there are these W and H functions, which I'll talk extensively about in a minute. But basically, these are the saturation and wettability distributions, and they act, as you can see here, to mix each of these T2 distributions to give a predicted T2 distribution. And in a perfect world, that predicted T2 distribution should look a lot like the measured T2 distribution that you've recorded for a mixed wettability, a mixed saturated sample. So now I'll just talk a little bit about these H and W functions. So these H and W functions are basically step-like functions which describe which pores are either filled with water, in the case of the H function, light blue shown here, or wetted by water, in the case of the pink function shown here. we chose to use the functions that look like this, and basically they're just step functions, and by changing, they only have one variable, which is RA, which is the position of the inflection point here, or RB for the case of wettability. So we basically can move these functions left and right on the T2 axis. We chose this function for simplicity, but of course you can make your function anything you want if you think it would serve your model better. But now it becomes clear on how the H and W functions mix the T2 distribution. So basically, by varying RA and RB, I'm sliding the H and W functions along the T2 distribution. So if I want to predict that my predicted T2 distribution has only a few pores entirely filled with water, then I move my RA function, my RA inflection point left or right until I have that. And that's exactly what I have here. And so in this case, I have the 100% water saturated T2 distribution, and I've put the H function to show that only these pores shaded in light blue are contributing to the to the water, or the pores that are filled with, that are wetted by water. And conversely, you can do the same thing with the wettability. So in this case, I put it over here, and now I'm saying all these pores are wetted, are wetted by water. So then once I've established what the right H and W functions are, they can then be used along with these equations here to predict the wettability. And very importantly here is this method also gives you a way to predict the brine saturation of your mixed wettability sample. And this will become really important. This is one of the keys to this method. Is it doesn't just give you a wettability. It also gives you a prediction of the water saturation in a mixed wettability sample. So this, because as you'll see, this becomes really important because it allowed us to fully verify our measurements, because we can always probably in some other method, whether it be mass or some other NMR measurement, determine the saturation. So we always have a way to double check that our model is predicting the correct water saturation. So, okay, so let's talk about how we apply the NWI method. Well, as I said, we basically mix the 100% water saturated, the 100% oil saturated, the T2 bulk oil, and the T2 bulk water T2 distribution. So we mix all these T2 distributions using the H and W functions that I just showed on the last slide. And so doing this, the output of the model is a predicted T2 distribution. So then if you want to get quantitative results and you've measured the T2 distribution of some mixed wettability saturation and you have a predicted one, then you can put this model in a least squares fit, and you can continue to predict T2 distributions until you've minimized the difference between the measured T2 distribution and the predicted one. And once you've minimized that, then you've arrived at your best fit. You minimize them. You continue to, prediction is always done by varying only these H and W functions. So you end up with a final H and W function after so many iterations through the fit. And now you've got a final predicted T2 distribution, which hopefully matches your measured T2 distribution very closely. So once you've done that, now you've got a final T2 predicted mixed wettability T2 distribution, as I said, and you have the final H and W functions. Then you can use those final H and W functions to predict the wettability index and the water saturation. And this is what we've done with our model. We do this fitting routine, and we've always recorded the 100% oil saturated, 100% water saturated T2 distributions from the same sample that I'm using to get my mixed wettability T2 distribution, and I compare this in this least squares method, and I'm always getting a new wettability index and a new saturation all the time. So I'm going to now try and explain this a little bit more clearly using some actual T2 data. Okay, so here's some T2 distributions for a conventional sample where I have the 100% water saturated T2 distribution shown in blue, the 100% oil saturation, in this case it was decane saturated, shown in yellow, the bulk decane shown in green, and the bulk brine T2 distribution shown in red. So now, as I said many times already, the name of the game is how do I mix the blue, the yellow, the green, and the red to get a T2 distribution that will look something like the brown T2 distribution, which was a T2 distribution I measured for a mixed saturation sample. To be specific, what we did for this sample is we had a 100% water saturated conventional sample, and we spun it in our centrifuge surrounded by decane and pushed water out and pushed oil in and ended up at a mixed saturation state. So here, this hopefully will become even more clear now. So here, going back to what I said about how the model works, I said it has 4 different fluids in the model. As I said, there are water wet pores and oil wet pores. So the water wet pores, where there's water on the surface of the pore, the T2 distribution should look like the 100% SW T2 distribution. And in the center of that pore, if I have oil, it should look like the bulk oil T2 distribution. And then for the oil wet pores, the oil on the surface of the pore should look like the 100% oil saturated distribution, and the water in the center of those pores should look like bulk water. So then I have a blue, a yellow, a green, and a red T2 distribution. And I measured those first. And now I've also measured the T2 distribution of this mixed wettability sample. So this is the sample I centrifuged and forced oil into. And I get a T2 distribution that looks like this. So now you can start to tease out, even in your head, you can start to tease out what the wettability probably is here. So it has this long T2 peak, which coincides almost exactly with bulk oil. So clearly, this is oil in the center of the pores. And then this T2 distribution is probably coming from the 100% SW. So this indicates that these are probably oil wet. This sample is probably water wet, but mostly saturated with oil. So let's see if the model says that. Okay, so here's the final data where we have the final H and W functions along with the measured T2 distribution for our mixed wettability sample and the corresponding predicted T2 distribution shown in black. And you can see here they agree pretty well together. But what more interesting here is I've plotted the final contributions from each of the T2 distributions. So you can see, as expected, most of this long T2 peak in the black predicted is coming from bulk oil shown in green. There is some contribution from 100% oil saturated T2 distribution, and then this smaller peak down here is coming from the water, the 100% water saturated T2 distribution. So as expected from what I said in the last slide, this is a water wet T2. It is water wet. It has a wettability index of 0.89, so close to 1 or water wet, but it has a low water saturation of only 13%. So the method worked. The method worked here. It gave the results as expected. But this was for a conventional sample. What about an unconventional sample? That's what we're here to talk about. So here's some more data, corresponding data, but now we're talking about an unconventional sample. And the first thing to note here is the T2 distributions are much more overlapped than they were for the unconventional sample. For example, the 100% brine saturated blue and the 100%, in this case, decane saturated T2 distribution were overlapped much more closely. And when you have more overlap, that makes getting a good fit more difficult. In addition, the bulk brine and the bulk oil T2 distributions were much more overlapped. But nonetheless, the model worked. The H and W functions that we arrived at were able to predict a good predicted T2 distribution that matched very closely the measured T2 distribution for a mixed wettability sample, unconventional sample. And in the final predicted water saturation was about 27%, and the wettability index was slightly water wet at 0.47. So the model worked. So that's good. So that we were confident after some initial tests that this could work and that we could track changes in wettability of unconventional samples. All right, so let's talk about some actual results. So in this study, we studied 6 twin core samples, and I'll talk about the reason we use twin plug samples in a second. But if you look, this is a table here, shows all some information on all the samples we studied. The core plugs spanned a variety of rock types from North American producing unconventional reservoirs. Some of these samples were really tight, and that made them challenging with porosities as low as 4%. As I said, these were studied as twins, and each twin sample came from reservoirs at the same reservoir at similar depths. And wherever possible, we tried to get extra information such as SEM images or helium porosity data, and in case this information might help guide us in our analysis. So what was our experimental procedure? So we decided the goal of this study was to observe the change in wetting condition of the sample as a function of time. And to do this, we decided to use spontaneous imbibition as the actuation method to cause a change in the distribution of wetting fluids in the sample. This is because other methods to try and force a change in wettability in these tight unconventional samples wouldn't be so easy to do. For example, using pressure to force fluids into the rock is very difficult with such tight pores and low permeabilities. So we had to settle on be patient and use imbibition to induce changes in wettability in these samples. So to do that, we took 2 twin samples. We would saturate one with an oil and one with a water, and then we would submerge them in the corresponding the other fluids. So for example, a 100, as shown here in this figure in the left, we have a 100% water saturated sample submerged in decane. We also would have at the same time a 100% oil saturated sample submerged in brine. And then we were just patient. We just waited, and over time we would remove the sample from the submersion fluid, dry its surface off, and record its T2 distribution. And slowly over time, as I'll show, these T2 distributions varied, and this variation was caused by a change in saturation of the samples along with a change in wettability. And this is interesting here. This picture shows some water droplets on the surface of an organic rich sample after it had been submerged in decane. So this is water that's been pushed out of the sample over time and bubbled up on the surface of the sample. And we would dry these bubbles off and record the T2 distributions. And these T2 distributions that we record over a function of time would always act as the mixed saturated distributions in the analysis I previously outlined. So we would have the 100% oil saturated and the 100% water saturated, the bulk oil and the bulk water T2 distributions on hand, and then every time we would take the sample out, dry it off, run its T2 distribution. We would use this mixed wettability T2 distribution as the one of the points in our fit, and we would generate a new wettability and a new saturation every time you removed it. And as you'll see, these experiments went on for sometimes months at a time, and that's going to become clear in just a second. So just briefly, what summary of our NMR spectrometer and the parameters. So the NMR measurements for this study were done on a 2 megahertz GeoSpec 275 spectrometer from Oxford Instruments. This is the one shown here. The NMR data acquisition was done using our advanced green imaging system, advanced systems software package. The data processing was done using our software along with MATLAB, and they were all the T2 distributions were recorded, as I said, with a CPMG pulse sequence, the parameters for which are shown in this table here. Okay, so before I get into some results, I just want to talk a little bit about the validation of this work. So this is really the key that made really the key to our work is how much validation we did. We wanted to be 100% sure that the wettability and the saturation data that you derive from this model were accurate. So we wanted to, I mean, in a perfect world, we would validate our measurements with a corresponding wettability measurement. But as I said, there's various reasons for not being able to get a wettability measurement of these unconventional samples easily. But what we can do is verify the saturation all the time. And one of the ways we could verify the saturation all the time is to repeat the measurements with D2O. As you all know, D2O is hydrogen NMR invisible. So if we repeat the measurement with D2O, we should only see the hydrogen in the oil, from the hydrogen from the oil. So this meant we should be able to determine the oil saturation directly from these D2O NMR measurements. And if you know the oil saturation, you can quickly determine the corresponding brine saturation. And this figure shows a typical T2 distribution recorded first where it was water saturated, shown in red, and you see two peaks. And then corresponding T2 distributions shown in blue. So now we're only seeing the decane. So you can see we repeated the measurements all with D2O. We only see the oil, so we could always at any time know exactly what the saturation was. So this is really the key to our measurements, is how much work we did to verify. And as you'll see, this was the only method of verification we did. We did other methods. I'll get to that in a minute. But in the next few plots, which are the key plots to our work, you're going to see a series of saturations and saturations derived from the D2O data, and how did they correspond with the saturations derived from the wettability analysis. So let's talk about that right now. Okay, so here is the key plot for two of our unconventional samples for our data. So on the left here, this is the wettability as a function of plotted as a function of time, and on the right here for the samples 7P3A and 7P2. And over here is the corresponding saturation predicted from the models in blue and red for the course for 7P3A and 7P2. So let's look at the wettability first. So it says that we started with this 7P2. It was 100% water saturated, and we submerged in decane. And over time, it slowly became more oil wet. Because correspondingly, for 7P3A, we had it initially, 100% oil saturated, submerged in brine, and over time it started as very oil wet, and over time it became less and less oil wet. And finally, by about 50 days, so close to 2 months, the wettabilities both saturated in this minus 0.5 oil wet region. And the wettability, that's the wettability. That's how the wettability changed. And for the saturations, it's similar. We started with this 100% brine saturated imbibing decane, 7P2, and very quickly, by about 50 days, the saturation stabilized near 30% water saturated. So most of the water was expelled from this sample. Correspondingly, for the one we started that was oil, that was 100% oil saturated, it became less oil saturated before stabilizing near 20% oil water saturated. So these agree pretty well, but the question is, are they right? And so we repeated the whole measurement again, but this time with D2O, and that's what the green and black traces show here. So the green trace is for 7P2, and in a perfect world, it would overlap perfectly with the red. It's slightly off, but it showed the same general trend. And similarly, the black is the D2O data. So we're driving the saturations directly from the NMR data, and we see it should, in a perfect world, match the blue trace. And it generally has the right shape. There is some discrepancy at early time. I'll have a little bit more on that in a minute. But eventually, they do reach similar saturation as the red and the blue, but it's not exactly the same. So the question was, are these discrepancies we're seeing between what the model predicts and what the NMR D2O data saw, does it really affect the wettability? So we went back and we went back to the NWI model, only this time we didn't make the model determine a saturation, we fixed the saturation at the known D2O saturation. And when you do that, you see this discrepancy on the order of 10% observed here in the saturation didn't have much change, didn't have much effect on the wettability. The wettability determined from either the D2O saturation in green or black versus the wettability predicted from the NMR data alone, all agreed pretty well and said this was a oil wet sample with a wettability X near 0.5. So this is good. This means that A, our wettability model is working, and B, the D2O data, if you have a slight discrepancy in your saturation, the D2O data, if you use that and use that to back out the wettability index instead, you'll get a number that's pretty similar. So this was really giving us confidence in our method. Here's another set of data for a different set of samples. These were samples GP2 and GP3. Again, let's look at the wettability first. So again, same as before, we start with one of the samples was 100% water saturated, imbibing decane, and another one was 100% oil saturated, imbibing brine. And over time, the two samples meet, the wettability meets, in this case, near 0.3, 0.4. The change in wettability here was much faster than the previous sample. So it seemed to occur much quicker. Within 10 days, it arrived at a pretty stable wettability. Well, what about the saturation predictions from the model? Again, one of the samples went from 100% water saturated to around 60% water saturated over the course of the experiment. Conversely, the one that was imbibing brine, 100% oil saturated, became around 50% water, 50% oil, 50% water sample over the course of the experiment. And what's interesting about this is there's some real oscillations in the data at early times. So these oscillations made it more important to repeat the experiment with D2O and see how the D2O measured saturation showed compared to the predicted saturation. As you can see, there is some discrepancy in the saturations measured predicted from the model versus those measured from the D2O. But as with the last sample, we went back and re-predicted the wettability. And whereas there is some discrepancy at early times, the wettability, whether it's predicted solely from the NMR data or it's partially predicted from the D2O saturation, it always arrives at, for this particular rock, a wettability that was around 0.2, 0.3. And keep these oscillations in mind. I have an explanation for those, which I'll get to in a minute. But again, this shows that even if there is some discrepancy and you go back and you fix the wettability, you fix the saturation at the known saturation, eventually you'll get a wettability consistent with the pure model number. Okay, so quickly, here's a slide of all the results for all 6 samples, all 6 twins we looked at. I'm not going to say too much about this. They had a whole different variety of, they had a marked difference in wettability among the sample types, and this suggests that a robust wettability quantification method, like the NWI method I'm presenting here, is especially relevant to for unconventional samples. And I would suggest that if you want more information on the specifics of these examples, please read our paper, appeared in Petrophysics in June 2022. It was a great summary paper of all our work, and it includes all the references to all the presentations we gave over the course of this work. And you'll see a lot of the figures that I'm presenting today were borrowed from this paper. So please, if you want more information, I encourage you to read that paper. Okay, so I'm going to talk a little bit more about even more validation studies we did to further validate our method. So, as I just said, in general, the brine saturations derived from the D2O measurements agreed well with the brine saturations predicted by the NMR wettability analysis. However, there were a few cases where there was more disagreement than we would have liked, and this is best shown by these two samples here, where 14.1 and 14.7. You can see there's quite a bit of disagreement between the saturation predicted from the model, shown in red and blue, versus the saturation shown from the decking. And especially for this 14.1, it's 14.7, sorry. The model really showed that the saturation didn't change over time, and that just didn't make any sense. So we wanted just an extra set of verification to make sure that the D2O data was the true saturation. And luckily we had recorded mass. So the purple and the brown here are a mass analysis. So the mass agrees with the D2O data, and it further cements that the D2O data is the real saturation, which we were pretty confident in already, and that it is truly, if you have the D2O data, you can always confirm the wettability, you could fix the saturation at the D2O value and then back out the wettability without any discrepancies. Finally, I'm going to talk about our final validation study, which was our 3D analysis. So one of the key assumptions to our method is that there is full and uniform penetration of the imbibing fluid into the sample. And so we wanted to confirm this assumption. We wanted to make sure that because our T2 measurement is a bulk measurement, we wanted to make sure that basically the imbibing fluid is penetrating the samples fully, so that our bulk measurement is a true measurement of what is going on entirely in the rock, and isn't just what's happening near the surface of the rock, and what's going on in the very middle of the rock isn't a reflection of what the T2 data is showing us. So we repeated the measurement with D2O. So for example, here's a 100% decane saturated sample submerged in D2O. We then took, over time, we recorded the 3D image of the rock. And over time, in theory, this image should get less and less bright as the D2O submerges into the rock. And I'll show a little bit more of this in a minute. But just briefly, the NMR data was recorded on a 12 megahertz geospec instrument by Oxford Instruments. Again, GIT software was used for data acquisition and analysis. All the images were recorded using the Sprite pulse sequence, the parameters of which are shown here. Okay, so what we did is we took the 3D image that I showed on the last slide over a series of times, and yes, they did get less and less bright over time. But I collapsed all the data down onto one XZ plane. So I collapsed along the length. So I summed all the data onto one two-dimensional image. So basically, it's as if I was looking top down on the image. I then looked at regions of interest within the rock. So I had the region of interest 1, which is the middle of the rock, region of interest 2, which is the next series out, region 3, which is just beyond region 2, and then finally region 4, which was just inside the rock. And notice I didn't include the very surface of the rock because I wanted to make sure I wasn't seeing any bubbles that might have been forming on the surface of the rock. So then I looked at the average intensity in each of these regions as a function of time. And ideally, this should show that over time, all these regions should see the same decrease in intensity. And this would ensure that, yes, the imbibing fluid is fully penetrating the rock. And that's exactly what I did here. So region 1, which was the innermost region, took the longest time to stabilize, whereas region 4, the outermost region, took the least amount of time to stabilize. But over time, over about 25 days, all 4 regions showed the same decrease, roughly the same decrease in intensity. So this showed that, yes, our method of using spontaneous imbibition to track the changes in wettability is valid, because we are seeing full penetration of the sample. But the other interesting thing this shows is that until about 3 weeks in, we do not have uniform imbibition in the sample. And this was recorded with sample GP2. And if you go back a couple slides back to here, and you see for sample GP2, where I had this oscillation in my saturation prediction method, and it didn't agree as well as it should have with the D2O data. Well, the D2O is a bulk measurement, so it's an average measurement. It smooths out any of these, any of this non-uniformity that is observed in the T2 distributions. And the reason for this oscillation, I now know, is because the T2 distribution that I recorded was not a true average of what was going on in the rock. Well, it was an average, but because it wasn't uniform, it wasn't an accurate prediction of what was going on in the rock. So what we learned is, if you're going to do these wettability measurements on these unconventional samples, you really need to wait 3 weeks roughly for the change in saturation to stabilize, and only then are the T2 distributions really reflecting what's going on in the whole rock. And anything before that, oscillations or disagreements between the model and the D2O data, is probably due to the fact that your inhibition hasn't fully saturated the rock yet. Okay, so I'll just conclude with a few conclusions. So we've shown that the NMR-based wettability index can be adapted to unconventional rocks. We've also found the NWMI model is well suited for data sets featuring complex oil and water T2 distributions with multiple peaks. Most importantly, we've completed an extensive validation work of our studies using D2O to confirm the oil and brine saturations. We have employed mass balance to verify the D2O data. We've used 3D imaging to show that our spontaneous imbibition method is valid. And we've developed a procedure. I didn't share it, but if anybody wants to do this work, we've developed a procedure, step-by-step procedure on how you should proceed if you want to do one of these NMR wettability T2 analysis. And you could see our paper for fully detailed discussion of this procedure. So that's the end of my talk. Before I conclude, I've just a couple of acknowledgments. I just first thank the SPWLA for inviting us to share this work. And most importantly, I'd like to thank my co-authors, Ron, Shana, and Dragon. This is really a collective effort on all our parts, between ourselves at Dragon and I at Green Imaging, and Ron and Shana when they were at ConocoPhillips. So it was a great collaboration. I enjoyed every minute of it. And Ron and Shana and Dragon, thank you very much. And I'm happy to answer any questions now.
All right. Thank you so much, Mike. Yes, if you have any questions, please raise your hand.